This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 531, recorded on January 18th, 2019. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Hello. Do you have snow?
Not yet, but it's supposed to be coming starting at 1 a.m. And 3 to 6 inches. Yeah, is what we're supposed to get.
Yeah, we had a little dusting this morning. I think we're supposed to get the same thing.
Yeah. Oh, now this other place says 8 to 12 inches through next Thursday. Okay, so over the next 7 days, we'll get a lot.
Okay.
But it's really our first winter, so we haven't had much to speak of.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. Forecast here is for 6 to 10 inches on Saturday night into Sunday.
Wovel time.
Yes, I'll be breaking out the snowblower and the wovel, I'm sure.
Cool. And back after a absence overseas from Madison, New Jersey, Brianne Barker.
Hi, it's great to be back. I'm finally back, although my brain may not be totally in this time zone. And we are, of course, going to have the same thing here, about 3 to 6 inches over the weekend. Our dusting's almost gone here.
Our guest today is from Pittsburgh, Pennsylvania. And that's where he is right now. He's at the University of Pittsburgh, coming back for a second time on TWiV. Pat Moore, welcome back.
Hello, everyone. Thank you very much for having me.
My pleasure. Last time, TWiV 160, more tumor viruses. That was a good one a long time ago. My gosh, time flies.
Yeah.
Well, good to be back. Have you back. We're going to talk about a recent paper of yours. First, we have a little business to take care of. We'd like you to fill out the listener survey. You can go to asm.org/TWiV poll to do that. It's very short, 5 minutes, 12 questions. Helps us to learn about you, maybe get some sponsorship. asm.org/TWiV poll. We'd like to get 1,000 people, and we're almost there.
Cool.
I want to thank the American Society for Virology for sponsoring TWiV. They will be a sponsor for this year. We're getting support from them now, in addition to the annual meeting that we traveled to. So we appreciate that very much. ASV, of course, is the Society for Virologists in America. There are lots of global virology societies. So this one is the American Society for Virology, with lots of members who love viruses, and they have an annual meeting. You can go to asv.org and check it out. And there's a meeting coming up, right, Kathy?
Yes, July 20th to 24th, 2019 at the University of Minnesota. And we do have a pretty good number of international members, certainly through the Americas, South America, Central America, but also Europe, Japan. I haven't checked the list of countries lately, but people from all over the world are in the American Society for Virology. Yep. Oh, and Canada. Sorry.
Canada, right.
Didn't mean to forget Canada.
There is a Canadian Society for Virology, but I believe they held their first meeting at ASV, didn't they?
That's right.
Right. And ASV has had a number of meetings in Canada over the years, and we will be there in 2021 in Montreal.
Yeah, we were at the CSV in Halifax, and they said they would prefer to have most of their meetings with ASV. Mm-hmm. But it's nice to have your own country version because it's a smaller community and it's different. It's a different purpose. But ASV is a big organization. You can meet a lot of people, and it's a lot of fun. So
it is, and I'm really excited about the list of speakers this year.
Yep. I also want to tell you about the European Congress of Virology, ECV2019.com. That is happening in Rotterdam. And the abstracts are due on March 1st. So if you're in Europe or you would like to go to Europe, what month is it? May? June? Let's see. April 28th to May 1st. And this, of course, has spawned on the last few episodes a little discussion of big port cities.
Yes. Rotterdam is a big port city, but it is not the biggest port city, we've determined.
And Kathy, you suggested that Piraeus might be pretty big too, right?
Yeah. Where did I find that? That was a Daily Overviews picture, I think. Yeah, that I forwarded to you guys.
Yeah, so Paris is right next to Athens, right?
Mm-hmm.
So that could be. All right, so that's the European Congress of Virology. Then the ASM Clinical Virology Symposium is May 5th through 8th in Savannah, Georgia. So that's clinical stuff. And registration is currently open. You have till March 8th to submit an abstract. You can go to asm.org/CVS. Now, last time I gave you the wrong URL, and it's asm.org/CVS. You should be able to remember that, right? Because you pass CVS all the time.
Right.
Okay.
The Intel International Science and Engineering Fair. It's the world's largest pre-college science and engineering exhibition. It showcases the world's most talented and innovative students, and it provides an opportunity for their work to be judged by professional scientists and engineers. So the next meeting is in Phoenix, May 14th through 15th, and the organizers need PhD and MD judges. They need judges in 22 scientific and engineering disciplines, including more than 200 scientists in the categories of biochemistry, cell and molecular sciences, biomedical health, and microbiology. We will have a link in the show notes where you could go and get more information about this and register if you're interested.
Cool.
There's a couple of outreach activities associated with it, and evidently it's really rewarding. Britt Glaunsinger told me that she's done it several times, and so I think it sounds really worthwhile. Check it out.
Now, a lot of you have complained that the beginning of TWiV takes too long, but, all this is public service stuff. It's things that people want to hear about, and we would like to spread the word since we have so many people listening. Unfortunately, we have some sad news. Marie Chow has passed away.
Oh.
Marie Chow was a virologist who I first met as a postdoc in David Baltimore's lab. And she worked on poliovirus and did that for many years at the University of Arkansas. And I just saw her last May. Unfortunately, went downhill quickly. However, on a nicer note, we have an email from Alice Huang, who wrote, Vinny, I have just sat through TWiV 528 and thoroughly enjoyed being brought up to date. That's, of course, our year-end review. I also concur with the feelings expressed at the end by the participants who feel that being part of what you do is a lifeline, especially for those of us in our retirement years. I'm glad to see that you included Raul Andino's work on RNA viruses in insect cells and the fact that they contain DNA copies of RNA viruses. I consider this work, along with that of Maria Carla Sally of the Viruses and RNAi group at l'Institut Pasteur, the most novel and interesting findings of 2018. I thank Kathy Spindler for remembering that defective viruses were long ago known as DI particles. I predict that these discoveries of defective viral DNA copies in insects and their cell lines will have an important bearing on mammalian viral infections as well. I wish you well and many Happy New Year's, Alice.
Yeah, I had a chance to talk to Alice in my office here. I think it was maybe early November. It was on a football Saturday. And that was really great. It was really fun. And, a lot of Alice's research was with defective interfering particles. So I'm sure that's why that resonated with her.
For sure. She was an early person to look at that. And you did a PubMed search. Mm-hmm. She has 26 papers on defective interfering particles. Yep. First one was 1969. Mm-hmm. Ribonucleic acid synthesis of VSV1, species of ribonucleic acid found in Chinese hamster ovary cells infected with plaque-forming and defective particles. And then in 1970, this is cool, a Nature commentary with David Baltimore, defective viral particles in viral disease processes. So they were speculating, are these going to be involved in viral disease? And now we know that they are. It's pretty cool. Mm-hmm. But as she says, she can't do any more research on them, but she can listen to us talk about it. Okay, thank you, Alice, for that. And now let's get to Pat Moore and more tumor viruses. Pat, you and Yuan Chang just published a paper, which is in PNAS. Circular DNA tumor viruses make circular RNAs. So you sent me an email a while ago saying maybe you could tweet about it. I'm only on anti-social media. I thought that was really funny. Did you start your own anti-social website?
Yeah, but it only has me on it.
It's only you, yeah. So this is a cool...
So I tweet myself, yeah.
This is a cool paper, circular RNAs, which have been around for a while and are getting really interesting in cells, and you found them in a couple of viruses. So let's talk about this. And the viruses in this paper are Epstein-Barr virus and Kaposi sarcoma herpes virus. So maybe we could start with a bit of a review of those and what they do in people.
Sure. So first, thanks everyone for agreeing to listen to us on this. So we sort of fell into this. This wasn't planned in any way, shape, or form, but like a lot of our research, just Yuan and I have been working together since we were at Columbia 25 years ago. And
you're putting some distance between us, huh?
It's certainly making me feel old.
Were you here?
Well, it was a long time ago.
Yeah, I was there when they were there.
Wow. Yeah.
And so we look at a herpes virus. Herpes viruses are large, double-stranded DNA viruses, and there are 8 of them among the human herpes viruses. Everyone knows herpes, right? But there are 8 different human herpes viruses, 2 of which cause cancers, and those are EBV and KSHV. And they're gamma herpes viruses. They tend to preferentially be latent in B cells, but also they can infect other tissues. And they're quite important because especially EBV causes nasopharyngeal carcinoma, which is a big cancer in Southeast Asia. It causes stomach cancer. So it's a quite common problem. And KSHV is also a big problem, particularly in Africa and in Western China, where it causes Kaposi sarcoma and some lymphomas. And then also both viruses are important in AIDS patients. So we've been interested in trying to find new cancer viruses. And we were keen on using a technique that we developed about 10 years ago that was successful in finding a polyoma virus that caused cancer called Merkel cell carcinoma. The virus is Merkel cell polyoma virus, and it causes most cases of Merkel cell carcinoma. But that's based on just simple sequencing of polyadenylated RNAs. And we tried that on a tumor that looks to us like it may be caused by a virus. And the tumor is called post-transplant lymphoproliferative disorder. So the reason why this is likely to be caused by a virus is because it occurs in patients, obviously, who are immunosuppressed from transplantation. And those tumors are mainly caused by EBV, but there's a significant fraction of them that are not. So we were interested in asking was there another virus that causes PTLD? So we sequenced those using this technique and couldn't find anything. So it's been sitting around for a number of years in our lab, 5, 6, 7 years. And we thought, well, let's see, maybe it could be a circular RNA virus. So really talented postdoc in the lab, Tuna Topton, working together with Bizu Aber, looked at these tumors and began to perform sequencing for circular RNAs. And how you do that is you first treat the RNA with RNase-R, and that'll begin to chew up all of the linear forms of RNA. And if you ribodeplete the RNAs, the ribosomal RNAs, and then sequence them, then you'll preferentially enrich for RNAs that are circular.
Was there any particular reason why you decided to start looking for a circular virus at this point?
Yeah, because we couldn't find one that wasn't circular.
Oh, that's a good reason.
So yeah, our idea was that, you know, if you have a tumor and it's caused by a direct viral pathogen that is expressing an oncogene in every cell, then it's likely to be made from a mRNA, from a polyadenylated RNA. So if you sequence the polyase isolated RNA at a sufficient level, you should, by random chance, come into that RNA, if it's at least at 1 copy per cell, which turns out to be, there are about 200 RNA molecules per cell, so something on the level of 200,000 reads or even 1 million reads, then you're, by random chance, going to sequence a viral RNA if it's present in each and every tumor cell. And when you do that now, you can compare that back to the human genome, subtract out all of the known RNAs that are aligned with the human genome, and what you're left with is a candidate pool of RNAs that belong to what we think might be a virus. And then we can look at each one individually. Well, we've done that for PTLD, and we got, as they say in New York, bupkis. So, that's famous, right?
Yeah, we say that here.
That was actually the word
right?
that was just coming to my head when you were saying we got.
We got bupkis. So that's the reason why we thought, well, you know, it almost certainly is a tumor in which the tumor cell antigens are determining whether or not the tumor will grow, and the immune response against those antigens. And presumably those antigens are foreign, and so they're probably due to a foreign virus. So we didn't find it. Well, let's look for other RNAs, see if possibly we could have missed it because it was a circular RNA.
So these circular RNAs, as far as I can tell, the first circles were viroid RNAs, right?
Right. And hepatitis D
and Delta
virus,
right.
Delta virus.
And then they were found in uninfected cells, right? Derived from cellular transcripts.
That's right.
So you were thinking maybe there's a Delta-like virus in these cells that might be doing this, despite the fact that none had been found, right?
Exactly.
Okay.
So if you're going to do that, then you also have to have a control. And here, in this case, Tuna used as her control a EBV positive PTLD.
Mm-hmm.
And by performing this circular RNA sequencing on the EBV positive PTLD, and then obviously we look at the EBV genome and see if any of these circles matched back to EBV, and it turns out they do. So she was able to identify circular RNAs that are made from EBV at specific sites. And there's some controversy about, or at least there's always been some controversy about circular RNAs, whether they're real or are they just a mistake in the transcriptional process, so that you just randomly get a circularization of an RNA, or do they have real functions? And the important part is that over the past 5 years or so, it's become quite clear that circular RNAs do have a regulatory function in eukaryotes, that they act as microRNA sponges. They can't, which means that they're antisense to microRNAs, and so they physically soak away microRNAs and inhibit specific microRNA functions. And they may encode proteins that are not cap-independent proteins. They can bind to RNA binding proteins. So there's a lot of things that circular RNAs can do. We just don't have any idea of whether they're doing that for this virus yet.
Mm-hmm.
But so that's the punchline to this, is that we found this from EBV, and then we obviously, because we worked so much on KSHV in the past, we looked at KSHV as well and found circular RNAs that are encoded by KSHV as well.
So this started out as a control for a completely different project.
Exactly.
And then you found this.
Right.
Which, of course, made it a very poor control.
Yeah. No, it wasn't a good control at all. But it's very interesting to us because, we've looked at transcription from these viruses, mainly KSHV, for years and years and years and decades. You had no clue that there were circular RNAs that are coming off of specific loci. And we're not the only ones who found this. Also, there's a large consortium of herpes virologists in the South, led by Eric Flemington and Rolf Rennie and others, Scott Tippett's in this group, and they also identified circular RNAs that were made from EBV, and Joe Ziegelbauer at NIH also found them from KSHV. So it was really nice that it's nice to be first, but it's even nicer to be right. And so, and I'm not even certain whether we were first. I think that Flemington's paper was first on this, but the whole point is that that a lot of people were looking in the same general direction for different reasons, and they find the same thing. And so that suggests that these circular RNAs are a new form of gene expression for these viruses. And particularly, there are at least one circular RNA for EBV and one for KSHV that are expressed during latency. And that really makes us perk up our ears, because it's during latency that tumorigenesis occurs for these viruses. And so it very well could be that these circular RNAs that we never knew existed until past year or so are driving the proliferation of cells, or maybe contributing to the proliferation of cells. But yeah.
So where are they coming from in the viral genome, and how are they formed? How do you form circles?
Yeah, so it's really interesting. I'm not patronizing anyone, but just to describe, that I think everyone here knows that that's listening to us knows that RNA is made first as a pre-mRNA copy from the DNA, and then it has a 5' cap put onto it and polyadenylation at the polyase site, and then it's spliced, and that's what Baltimore and Roberts got Nobel Prize for.
Phil Sharp.
Phil Sharp. Yes.
He'd be very mad.
Yes. You did it in Baltimore. No, you did it in Boston. That's what Sharp and Roberts looking at adenoviruses, as a matter of fact. So viruses are important. They were able to show that the RNAs the pre-mRNA is processed, spliced out, and so you have this discontinuous piece of RNA that is religated back together. Now, if you think about that, there's a donor and an acceptor site for any intron that is going to be spliced out of RNA. And if you have multiple introns, then if you have a downstream donor site, that instead of it going on to splice to its downstream acceptor site, if it wanders back upstream to target a upstream acceptor site, then it will form a circle. And that's precisely what happens. That's one way that circular RNAs can be made. And also, there's some evidence that there's various structures like ALU elements, things like that can bring RNAs together, and they then can be cut and religated to form circular RNAs. So
is that the phenomenon that you talk about in the paper known as backsplicing? I hadn't heard that term before.
So backsplice.
I had heard the term, but only in a nautical context.
Well, I think in a nautical context, you don't want to do backsplicing, but maybe you do. But so backsplicing, what that refers to is forward splicing, of course, occurs when an upstream donor site targets a downstream acceptor site and splices out an intron. Backsplicing refers to that upstream or that downstream acceptor site wandering off and then binding to an upstream donor site. And so you can imagine if you have a piece of RNA and it's just like a piece of string, if you take that string and put it together in a circle where the two ends come together, that's a backsplice. And so one way that you detect circular RNAs is you can do divergent PCR, which is RT-PCR, where if you take the very ends of a piece of RNA and are going outwards, if that is now put together as a circle, you can see that they would cross the junction, the backsplice junction, and would amplify. And so, whereas if it were a linear piece of RNA and it was turned into cDNA and then amplified, you would get no amplification because the polymerase would run off the ends of the molecule. So being able to amplify across this backsplice junction is one way of detecting circular RNAs. Now, because most RNA most RNases are exonucleases, and that they tend to chew up linear RNAs preferentially.
So exonuclease meaning they chew from the end.
They chew from the end, particularly from the 3' end. And so that what happens is that RNA is very unstable generally. So I had one of my first teachers, and when I was sitting in a class in Berkeley, he was telling me about RNA nucleases, and he was describing how he would isolate RNA, run it out on a gel, and he could watch the band disappear as it ran down the gel because he was contaminating it, and the RNA was being degraded. So, and that was just simply because, gloves, didn't wear gloves, things like that back then. And so the issue is that these circular RNAs can be highly stable, not necessarily highly stable in cells, but they can be highly stable, so that we've gone back to a Kaposi sarcoma tumor that was, a patient had Kaposi sarcoma early on in the AIDS epidemic back in the 1980s. And this is a paraffin-embedded tissue. So as probably you all know, the formalin and paraffin-embedded tissues really destroys the DNA, and you have to try and repair it and things like that to be able to get a PCR amplification. So we did this divergent PCR for circular RNA for the KSHV circle, and it was positive, even though this is a 30-year-old tumor that's been sitting on
formalin-fixed, paraffin-embedded, yeah.
Room temperature. It's been sitting in our lab for 30 years almost. And we could not even amplify any cellular DNA from this. So that has potential maybe for being a diagnostic. I don't know whether it will be or not, but I mean, that's one immediate consequence is you can think, well, perhaps we can detect these circular RNAs as they're shed from tumors into blood or into urine, and that might be useful.
Oh, if all RNA were circular, it would be so easy. Now, what is an endo that would cut a circle? Do you know?
Yeah, RNase H will.
Okay.
If you have a DNA,
yeah.
You have a little piece of DNA, and that's So the technique that I described, this RNase R sequencing, gives you a lot of sequences, and there are circular RNAs that come out, but many of them are just linear RNAs that are highly resistant, like leftover ribosomal RNAs and things like that. So you have to, or you should, test to make sure that the RNA is circular. And one way to do that is using RNase H and a DNA primer that goes across the backsplice junction. So that primer, if it's, you only have 10 base pairs on either side of the splice site, it won't bind very well to anything except for a circular RNA that has that junction. Then you can add RNase H, and if it disappears, then that's very strong evidence that it actually is circularized, or at least it has that junction.
Now, when you originally did this sequencing, you subtracted out all the cellular sequence, and so you got rid of the cellular circles, right?
Nope.
They're in there?
Didn't.
They're in there?
They're in there. Yeah, they're in the sequencing. But we subtract them in the sense that we used the EBV genome to align using it, because you also have to use a program that we use a program called Siri2 that identifies these backsplice junctions by finding a pattern sequence that is going in the opposite direction and suddenly stops at a certain spot. And that's what would happen if you had a backsplice junction from an RNA.
Speaking of programs, I love one of them that you used called Human Splicing Finder 3.1.
Yep.
It's great.
Splicing humans. Yeah.
I looked that one up because I wanted to understand the term high entropy score, what that meant. So,
gosh. Did you find out? Because to be honest with you, I'd have to go ask Tuna what that means.
No, I didn't really. I mean, it didn't really relate. Well, I looked up high entropy and I got that, and it was a bunch of words that mean something in English, but together, as it was written, didn't tell me anything. And so
that's common in our papers too.
No.
By the way, Dickson has arrived.
Hello, everybody.
Hi, Dickson.
Hey.
Hi, Dickson.
How you doing?
Are you out of breath?
No, I'm out of ideas, but I'm not out of breath.
All right, see you later.
No. I get to express those ideas.
Okay. Now, Pat is talking about circular RNAs.
Yep.
Okay, found in cells infected with EBV and KSHV.
Exactly.
That's to bring you up to speed.
Indeed.
Have you ever, Dickson, does any virus have a circular RNA?
They do not.
Does anything virus-like have a circular RNA?
Not that I've ever heard of before, but today it's different.
How about viroids?
Okay.
Right. There you go. Viroids invented them.
Well, okay, fine.
All right. So, Pat, you were able to map these circles to specific parts of the virus genomes. Tell us a little bit about that, right?
Yeah. So, for EBV, the part of the circular RNA is located in a region called the BART. In fact, it has a even better name, I think. They named the EBV genome by first BAM restriction, a digest of the genome. Then whether it was a rightward or a leftward gene, and then A, B, C, D, E, F, D, da, da, da. So you'll have names of genes like BALGA1 and BALF5. But this is, it is BARF. And since it wasn't found originally, they called it zero. So it's called BARFO.
Yeah, I remember that.
And it turns out that that's a very important locus because it wasn't, the fact that tumors were expressing viral proteins that were found, EBNA1 and EBNA2 and LMP1 and these other viral proteins that look like oncogenes or oncoproteins, and they do act like oncoproteins. They drive proliferation of these cells. Couldn't entirely explain how certain cancers arose. And it turned out that there are other non-coding RNAs that are made. Some of them, for instance, EBER1 or EBER2, which are used for diagnostics, are very highly expressed. But then finally, it was found that this Barfo region by Nancy Robb Traub's group, I believe, was expressed. And later, it turned out that one of the this is a highly spliced gene, and one of the introns makes all the microRNAs, or most of the microRNAs that EBV expresses. So that's a long prelude to say that because this is a highly spliced gene, it turns out that one of those splicings puts together a circular RNA. And we have absolutely no clue what it does, but it certainly is being made in the right place, and it's being made in the right way to do something interesting in terms of causing cells to proliferate.
Can you try to interfere with it with a reverse copy of the circular RNA? Try to silence it?
Well, yeah, we're this is Dickson?
Yep.
Hi,
the non-virologist, Pat.
No. No more than I do. So the other non-virologist on the I was trained as an epidemiologist. That Dickson knows that. In fact, I was in the School of Public Health.
Indeed.
That's why you know each other, right?
We do.
Yeah. So we were just talking about you can put in DNA, and you can actually transfect DNA and get the same effect of it'll match to the backsplice junction, which is unique. Now, remember, all of these RNAs are also obviously perfectly encoded with the linear RNAs.
Right.
And the only difference is that two ends are put together so that you have to find a way, and that's why they're kind of hard to work with, is you've got to find a way to distinguish the circular circles from the linear RNAs by looking just at that junction. And if you do that, then the RNAs tend to get cleaved and processed, and you can see whether
I got a little lost on the genes here. Is Barfo the same as BART?
Yes. Okay. Yes, I'm sorry.
So when we got to the point,
most people call it BART, but I like the original name is BART.
Oh.
So I think
so. You don't get sick on the the Bay Rapid Air Transit Authority. Okay, fine.
Barfo on the BART.
Exactly right.
That's right.
So maybe you're going to get to this, but I like this figure that you have where you show the gel with the 4 different sized bands, 700, 600, 500, 400, and then you indicate from each of the bands what the structure is. It's just a brilliant figure. And
that's not me. I'm thinking
for whoever in the group did it, and you approved it. But yes. But because of that, it's really clear that some of these still do have an intron in them, and some of them don't.
Right. And
so I was wondering if the presence of the introns suggests an order for this process occurring, or are they dead ends no matter what? And if they have an intron in them, then that's the way it is, or could it still yet to be spliced out?
Yeah. So so there's a couple of interesting things about these RNAs. Is that first of all, there are these circles that have retained one intron. And I have the opportunity to retain more than that, but there's only one intron that they have retained. So for whatever reason, the pre-mRNA, when it goes back and backsplices, it has a choice of either taking out the intron from the circle or taking out both introns, and only takes out one. So I don't know why. And that also means that the circular RNA has a unique sequence that wouldn't be found in the mRNA, which is the intron. So if there's something that the intronic sequence is doing in terms of binding to either a protein or another nucleic acid, you have that intronic sequence that could possibly do that. Now, the intronic, so our audience has to imagine, because they probably don't have the paper in front of them,
Although the paper is open access, so they can pull it up if they're not driving.
They certainly can.
That's right.
And if they are driving, it's going to be a very short read.
Yes.
You have to get off at the next exon to read it.
Ooh,
That's if you're out of gas. You can stop at the exon.
good, Dickson.
Sorry, Pat.
No, that was good.
By the way, that's why he left.
So you were describing the figure.
But I didn't give far enough away. I'm describing the figure, and so you have to imagine that this is a RNA that has multiple splices, and so it has exons to it, and then it has one intron that either can remain in it or not. And there are two flavors of that. It can start at one backsplice junction or the second. Now that's kind of complicated, but it's what I'm trying to say. But just suffice to say that there are two backsplice junctions, and they form 4 different RNAs depending on whether the intron is retained or not. Now, if you retain the intron, obviously the RNA is going to be larger than if the intron is excised. And it also turns out that the intron-containing RNA is retained in the nucleus, whereas the other RNA looks like it's either in the cytoplasm or distributed in both the nucleus and the cytoplasm. Now, could there be a function to the intron-containing RNAs? Yeah, and it's interesting. We just found this and just published this descriptively, so we don't have a mechanism, we don't have a target, but there's a lot of things to do.
Yeah.
These would any of these have an AUG in front of an open reading frame that could be translated?
Well, we looked at whether they could be were translated by rather than by sequence. We performed a polysome fractionation. So that's where isolate the ribosomes and isolate the RNA from different fractions, and you can actually tell whether the RNA is being translated or not. And we didn't see any evidence that there is a protein being made. It was just the first go through one.
Absence of evidence.
So I would say you need a positive control where you have a circle with an open reading frame and show that it does associate with polyribosomes, right?
Exactly.
Because I don't know.
Exactly.
I'm not sure how often they do. Okay.
Yeah, that's a good point.
Now, you have, if you take EBV and infect a cell in culture, do they make these circular RNAs?
Yeah. The interesting...
Because the ones you got were from tumors, right? Human tumors.
Well, yes. There is a form of EBV that is probably the most common laboratory strain called B95.8 or -8. And that was a strain that was originated by George Miller in marmoset cells, maybe in the 70s or 80s. And they're commonly used for instance, immortalizing B cells from patients with rare genetic disorders so you can maintain the DNA stocks. So a lot of people are working with that. But it turns out that strain of EBV has a deletion for both the microRNAs and for the circular RNA region. So what that's saying to us is that in the artificial circumstances where B95-8 is transforming a healthy B cell into an immortalized, maybe cancerous B cell, it doesn't need either of these circular RNAs or the microRNAs. It doesn't absolutely require it. But so that's an argument against them playing a role in cancer development. But it is the only strain of EBV that has that mutation that I'm aware of. And it is likely that in the natural setting of an EBV infection or an EBV-derived cancer, that it very well could require both microRNAs and the circular RNAs.
But that strain, can that undergo lytic replication?
Yeah. Mm-hmm.
So
you actually can harvest the virus and then infect B cells, which is how the immortalization is done.
Okay. And as far as you can tell, this is the only part of the EBV genome that gives rise to circles.
No. This is the region that is most abundant, but there are, I don't know, Eric Flemington's group has looked at this quite extensively, and I'm not certain how many, 10, 20 different sites are found. We found circles from a gene called LMP2, BHLF1, which is interesting because Paul Lieberman, who works on replication of EBV, has shown that an RNA from BHLF1 is involved with regulating the replication of the virus, but they couldn't quite define what that RNA was. And so one potential candidate could be the circular RNA, could be in some way determining whether or not the virus is able to undergo lytic replication.
Mm-hmm. What fraction of human tumors have these circles?
Well,
for EBV anyway, since we're
for EBV, all of the EBV tumors have them as far as we've tested. So we've tested nasopharyngeal carcinoma, PTLDs,
Burkitt's lymphomas. We've tested a tumor that is an oddball tumor, an interesting oddball tumor that we, working with Ethel Seserman at Columbia, first defined, and this was a B-cell lymphoma that has both KSHV in it and EBV in it. It's one of very few tumors that has two different cancer viruses in it, and they're both probably important for driving the proliferation. Certainly we see the circular RNAs in that tumor. It's called a primary fusion lymphoma, or PEL, when they're infected with EBV. And so a very analogous, it's not identical, it's analogous situation occurs for KSHV, looking at Kaposi sarcoma tumors and looking at multicentric Castleman's disease and primary fusion lymphomas. But you didn't find it in every tumor, right?
No, we didn't, but I think that's likely to be a case. We certainly can see it in every cell line, which is the lymphomas. We didn't see it in the original tumors, but that could be, preparation of the tumor. When you're looking at patient tumors, sometimes even the tissues that you get and you cut them, they may miss having tumor in them, or the tumor may be degraded. Even though that one case we did see the circular RNA that was retained for decades, we don't see that commonly. So we can see in about 30% of Kaposi sarcoma tumors that we've looked at, we can find the KSHB circular RNAs, but I strongly suspect that natively in the actual tumor that they're actually being made.
Does that suggest that this is a host characteristic rather than a virus characteristic?
Well, I my guess is that they're doing something to the host for the benefit of the virus. I mean, viruses are funny that way, aren't they?
In a humorless way. Yes. But what I was driving at was that how do you get RNA to make a circle? Do you ligate it, or do you does it just spontaneously form?
Well, you weren't here.
I wasn't here for that. I'm sorry.
Well, but that's okay. Repetition is a key to learning.
Yeah.
So again, how this occurs is it's a splicing phenomena,
right,
where a donor site for a donor region for an RNA will, if an RNA has multiple intron exons, a downstream donor site can reach upstream and target an upstream acceptor site.
Okay.
And then if you can imagine that, it would be like putting two ends of a string together and you'll have a circle now.
Sure.
Right. And that's the backsplicing.
And that's the backsplicing.
Right.
Which in the nautical context would be called an I-splice, but
They have to
Is that right?
They have to do with tying a
A
knot.
bolin.
well, an I-splice makes a loop in the end of a line.
I see.
Oh, so maybe those are good. I
But
don't know.
backsplice, in the RNA context, it's called a backsplice to distinguish it from a forward splice, which is going to the
Downstream.
normal or the downstream splice site.
But with respect to your question, Dickson, Pat, have you looked for the sequences in these circles in the cell genome?
Have we looked to see if these are
Represented in the cell genome, in other words?
Yeah, and the short answer is no, they are not. None of these regions know that both by that we know that KSHV does not have a unique match to that. Now there could be sites within the circular RNA that are homologous and could align. So I'm not saying that they do not align to human RNA or DNA sequences, but the entire circular RNA is not represented in the human genome.
Because one possibility is that they are sponges for cellular microRNAs. Right?
Oh yes, absolutely. No, I agree with what I mean. Yes, the answer is that there could be short regions that could be microRNA sponges, for example. We have looked for both BART and VIR4, and we're unable to see a microRNA interaction with those circular RNAs. But that doesn't mean it isn't happening again.
Sure.
Do the circular RNAs from EBV and KSHV have any similarities in terms of sequence?
Not in terms of sequence, but if you how should we? They are analogous. So I told you about the KSHV VIRF circular RNA, but there's also another circular RNA that is a very strange locus in the virus. This locus is called PAN, which stands for polyadenylated nuclear whatever. PAN. So
it's next to Peter.
So I guess you have to say good one. That was really dumb. So the PAN RNA it's an unspliced RNA. It's polyadenylated. And it can represent, and it's under extraordinarily strong promoter, so that it's mainly expressed when the virus undergoes lytic replication. And it can represent 80% of all the RNAs that are made from the virus during lytic replication, just that one little stretch of polyadenylated RNA. And it's not turned into a protein. We don't know what it's truly doing. There's a lot of speculation about it. And a lot of really interesting speculation, a lot of good work on it. But it turns out that what we see is that circular RNAs are made not only from the sense strand of this RNA, the PAN RNA, but they're also made in the antisense strand. Now, the PAN is not spliced, so how are these circles being made? We don't know. And so what we see is that it looks like a swarm of no individual circular RNA is very abundant, but just a whole swarm from this region in both directions are being made. And PAN, if you add TPA or sodium butyrate, which induces lytic replication of KSHV, you'll see PAN RNA, jump up, like I said, up to 80% of the RNA, total viral RNA that's being made. You'll also see these circular RNAs jump up in their abundance, whereas the other circular RNA I was talking about, called viral IRF4, circular VIRF4, actually goes down when the virus initiates lytic replication. So it's quite curious. We don't know. I've told you the story that sure we know exactly how circularization occurs. That a downstream donor site invades an upstream acceptor site, but that's not really true. We don't have any clue as to what the actual sequence features are that are causing circularization, for instance, at PAN. It's just a swarm of circular RNAs at different sites.
So if you made a preparation of just the circular RNAs and then micro-injected them into a cell, do you think it would have any effect on the biology of that cell?
Well, that's the kinds of things that we're trying to do, but it's actually better there are ways you can make exogenously expressed circular RNAs. There's a gene called lacase, for example, that has 2 repeat structures that take an RNA and essentially loop it back onto itself, and then it tends to cleave at a specific site. And it's been sort of slow going because that cleavage process, you have to get it just right, and you can't necessarily predict when you insert your gene into the lacase operon, you won't necessarily see it recircularizing at that backsplice that you want to see. So we've been mutagenizing this, but now we have them, and it looks like we can just take this gene and now express it in cells, and it will make a circular RNA of the circular circles that we want to look at.
No, it's good. Nice.
Cool.
Very cool.
That's really interesting. I think I was thinking of something similar to Dickson, where I wonder if any of these are recognized by cellular RNA sensors, or whether this is something that could, function as a sponge, but also avoid RNA sensing.
Right. So
it sounds like they're structurally similar to the cellular circular RNAs.
Oh, yeah. Well, there's a story that there's a story from a group at Stanford that found that if they took a circular RNA, this is, I believe, a cellular circular RNA, expressed it in cells, so it was made and circularized in cells, that it would not provoke any RIG-I signaling responses. But if they made that same circular RNA exogenously and transfected that in cells, then it did. And so it suggests that the RNAs are being recognized separately, and that there could be RNA sensors that are recognizing circular RNAs or something about circular RNAs. It's all kind of not known, but interesting that that they could be activating an immune response. So that also suggests that, who knows, maybe these are interfering with that response, so that certain innate immune signaling pathways are turned off. But that's pure speculation. We don't have data on that.
Are any of these packaged in virus particles?
That's... Well, if we had more postdocs, so this is a good place to. But yes, we finally made purified virions with help of S.J. Gao here at Pittsburgh, and we're looking now as we speak to see if they're packaged.
Yeah.
And it turns out that Tom Schenk first found this with CMV.
Right.
Is he found that herpes viruses, which are DNA viruses, are actually also RNA viruses, because they make RNAs, viral RNAs, and then they encapsulate those viral RNAs in the virion, and so they microeject these preformed RNAs, mRNAs, into cells as they infect cells, and can target innate immune pathways, for example, right when the virus enters the cell.
How do they know to do all this?
No.
This is gotta be.
No.
How does this work?
It's called evolution.
It's selection, nice.
Mutation.
Nice.
So, all right, virus particles. So the other thing you looked at in this paper is cellular localization. What's the significance of that?
Well, mRNAs are generally pre-RNA is in the nucleus, and then you're talking about the nuclear versus cytoplasmic localization.
Yes, that's right.
Yeah, so the pre-RNA, of course, is localized to the nucleus. It's processed into mRNA, which is exported, and then proteins generally are made from it. So what we found is that there was differential retention in the nucleus of the circular RNAs, whether or not it could be just size, could be just that these circles are too big to be exported from the nucleus, or it could be that there's something specific about retaining an intron in these EBV-containing circular RNAs that makes them stay in the nucleus and have some function.
Yeah, viruses can handle that, right? Because most of the herpes simplex transcripts are not even spliced, right? So they can figure out how to get out. So that probably doesn't block it. Maybe something else.
Right.
Now, you mentioned that you might be able to make diagnostic tests. We talked about how stable they are, and paraffin, formalin blocks, and so forth for 30 years. But what would you need to know before you could actually do that?
Well, we have to, all of the diagnostics have to be based on that backsplice junction, that unique part where the ends are together. And the nice thing about that with divergent PCR is you can just simply take a tissue, whether it's serum even, free. It doesn't need to be cellular necessarily, because of course there's a lot of diagnostics, cancer diagnostics. What they call a liquid biopsy is to look at DNA that's released from tumor cells in a patient and to be able to identify it and sequence it. So one may be able to do that also, and one would hope that it would be retained and would be more stable than the DNA even is, and it very well could be, so that you could then take that and detect it by RT-PCR. That's one example. There's another technique that we've actually used for looking at the circular RNAs in tissues, and it's called BaseScope. And it's a very interesting technique. It's hard to describe, but it takes two primers next to each other, and it builds an amplification structure that then has a chemical change, and you can see staining in the microscope. But it requires these two primers to be perfectly aligned next to each other, which is just perfect for a backsplice junction. Make one primer on one side, one primer on the other side. And unless those two primers are put together in the RNA, it will not amplify. And we can do that, and we can see some staining in cells and tissue culture. So it works actually beautifully for EBER, which is not a circular RNA, but it is an EBV encoded RNA. So that would be possibly useful for diagnosis in tissues. Now, there's an interesting story about EBV, and that's with nasopharyngeal carcinoma. There's a physician in Hong Kong, Dennis Ho, who's really well known for his work on detecting, for instance, fetal DNA in maternal blood and things like that, which you can even use for pre-genetic diagnoses or pre-birth genetic diagnoses. He has looked at serum from patients, huge number of patients, 20,000 or so, in Hong Kong, just coming into the emergency room and took their blood and then performed PCR for DNA for EBV. And then he waited some period of time, and those patients then developed this cancer, nasopharyngeal carcinoma. And he went to look and to see how frequently those patients who came into the hospital first had high amounts of EBV DNA in their serum, and he found that it was very predictive. You could see that if you were to take serum from patients in Southeast China, it would be a really good screening survey. So that's the kind of thing that may work for circular RNAs as well. And so those are some of the things that we're thinking about. They may not turn out to be, none of them may turn out to be valuable, but nonetheless, it's interesting and keeping us busy.
But in this study you just talked about with looking for EBV DNA in serum, what at the first glance when they first come in, what fraction of people have EBV DNA in their serum?
Well, nearly 100% of adults are infected with EBV. So we all have it somewhere in our B cells, but whether or not if you take, say, a milliliter of serum and then isolate DNA from that cell-free serum and actually detect DNA from that, would you see it? No, generally not. However, patients that have a tumor are shedding EBV genomes as their tumor cells die.
I see. And
that's cell-free, and that's at some level in their serum, and then you can detect it by PCR.
So if you are infected, if you have EBV in your B cells, which you say most of us do, I guess it's not always producing virus, right? It's periodically reactivated?
Yes. So it turns out that roughly 1 out of 100,000 B cells in your body are circulating B cells, on the order of that, 10 to the 5th, 10 to the 6th, are going to be harboring EBV, and that's primarily in memory B cells. And the same thing's true for KSHV in those people who are infected with KSHV. But for KSHV, in America, it's generally less than 1 to 2% of the general population is positive. But in parts of Africa, it can be 70, 80, 90%, just like EBV. And then you see a very similar thing is that if you take B cells from patients, you'll find the virus, but not necessarily in their serum where it's cell-free.
Right. So even if it's reactivated in a B cell, the virus is going to be confined. It's not going to be in the serum unless, as you say, there's a lysing tumor. Wow.
It's very, very limited. And, obviously it's going to be there at some level, but you just can't routinely detect it.
Yeah, sure. But on the other hand, if you look for antibody, would you find it in serum?
Oh, yes.
Higher. Okay.
And that's how we know that over 95% of people are infected with EBV.
Mm-hmm. Now, you mentioned in your paper at the end, what about other viruses? I'm sure people are looking, right? What do you think? Is this going to be in every DNA virus?
Well, the one thing that's interesting about circular RNAs is similar to microRNAs, is that microRNAs were first found by Tom Tuschl's group at Rockefeller in EBV and KSHV, and then they were found in polyomaviruses, SV40, and so it seems to be more common now. We found them in these two viruses. They certainly are in the rest of the herpes viruses, so that, in fact, Eric Flemington's group has published on the mouse analog to KSHV and the rhesus analog to KSHV, and they can identify circular RNAs in it. Now, do other viruses, adenoviruses, any other viruses have it? I would bet yes. But the thing is that we didn't see these circular RNAs until we specifically looked for them.
Sure.
So you have to look
Well, a paper like this and the others that you mentioned, they're going to stimulate people to look.
A lot of people are going to go looking.
For sure.
Yeah.
And you should also look in RNA virus infected cells.
Absolutely.
Because some of them splice. And as you said, some of the mechanisms don't seem to involve splicing, right?
Right. And we have an example of that from hepatitis delta. So that's circular. And I mean, it's a weird virus, weird and bad virus. But
that virus probably thinks you're weird too. You know,
I think all viruses are weird.
So Delta is the only, as far as I know, the only circular RNA element that we know of. It's a satellite. You know, as I said, they're in plants, but it's the only one in eukaryote in mammalian cells. But there may be more out there.
Right.
Of course, the whole genome is a circle. That's really cool.
Yeah.
Right? It doesn't mess around with backsplicing it. Hey, I'm just making a circle, and it's really stable. And of course, it has to be packaged in another viral particle, hepatitis B virus, because it doesn't encode a capsid. It's very cool. These plant viroids, by the way they seem to cause disease, they get into a plant cell, they're chopped up, and they're basically chopped up into microRNAs that target plant genes and make them sick. And one of them is the chlorophyll gene that makes the leaves all chlorotic, right?
Oh, wow.
By making the virus into a microRNA.
You know a lot more about this than I do. That's see, it's good. Yeah, I need to listen to TWiV every week, and I'd learn something.
That's what happens when you do TWiV for over 10 years. You learn stuff.
It doesn't happen to everybody.
Well, I just really think viruses are pretty neat. And as Dickson say, they bring joy to my life.
They do.
Except when I get a sore throat, then. Then they don't bring so much. So, Pat, you mentioned a few things. What else are you doing? Are you going to just forget this and move on to other things, or are you going to pursue it?
Not if the RO1 has anything to say about it, right?
Well, yeah, that's exactly so. But now we're down at the cutoff level. I just heard for NCI is 8th percentile. So
is that a good thing?
It's approaching zero.
It's approaching zero.
It's asymptotic.
Well, what that means is you should do your research because you enjoy it, you think it's interesting and beneficial to humanity, and to hell with getting funded for it. So there's what I think. But
science like nobody's funding. Yeah.
That's right. Exactly, like nobody's funding. So what are we doing? Well, we're continuing to look for new viruses, new cancer viruses. We have a very long project to use proteomics to find new cancer viruses, and that's really interesting. Can't tell you anything about it, but we've been doing it for 10 years, and we'll probably be doing it for another 10, but it's good. And then also we've found this other virus, this Merkel cell polyoma virus, has an interesting feature of targeting cells and arresting cells in mitosis. And when it does that, what Masa Shuda in our lab, who's now a assistant professor here at Pittsburgh, what he found is that it changes the cap-dependent translation. So these are all things. Transcription I know nothing about. Translation I know nothing about. But I have to learn it again. And in fact, as Vince knows, is when Yuan and I were first starting out working on KSHV, 1994, we'd go over to him and Saul Silverstein and say, "What is this RNA stuff?" And so
that makes me feel better already, by the way, Pat. Thank you.
So they taught us. They said, "Well, this is what it is. This is what it does." And so it's just something new that we're embarking on, and it's a lot of fun.
I still remember the days I used to see you and Yuan come to see Saul. You didn't come to see me at all, but Saul was right next to me, and you guys would walk in and wave and go into his office, close the door for a couple of hours. Yeah.
And I'd be sitting in the lobby in the little sitting area there eating pizza probably.
Oh,
Yeah, it was different back then. Now, one thing I wanted to note here on this paper, it tells you who reviewed it.
yeah.
Is that an option
yeah.
now on PNAS?
I think that this is a contributed paper. How this works?
Oh, okay.
I think
Right.
what they do is folks who are NAS members,
Right.
you can send in 3 a year.
Right.
And it used to be just send it in and essentially spelled things correctly, and you got to publish.
That's right. Not always.
They're tightening it up considerably now, and so you can identify who can review your papers, but first they publicly have to say, I reviewed this paper, so it doesn't look like it's
I see.
quid pro quo or there's any funny business going on. And then also for a contributed paper, boy, if you don't agree with anything, you have to justify it. I mean, it's the editorial board is tough. So I think PNAS really, most of their papers are people who are not at any association with the National Academy, and so they want to maintain that high level of rigor, and they're not treating a paper that's coming from an Academy member very much differently from anyone else's.
High level of rigor mortis, did you say?
High level of rigor mortis.
A lot of a rig eye.
So,
They have a lot of riggers to do all the splicing.
if this, man, out of control. If this,
Let's talk about chickens.
if you send, Dickson wasn't here for the chicken part. If you send a paper in not as a NAS member, you get the normal review. They don't put their name. The reviewers don't put their names on it, right?
I think they give it an option, but to be honest with you, I don't know.
Yeah. Okay.
But I think that's true.
I know Saul always used to sign his reviews. He said, "I don't give a damn if people know that I don't like their work."
Those may not have been the exact words that he used.
No, they were not. But this is a family show, right?
Yes.
Well, I did that too for 1 year, and my postdocs hated me for it because they obviously thought that they would take the brunt of that, and so I stopped doing it.
Yeah, because they figured people figured they were writing the reviews, right?
Right. They're writing the reviews, and that's a terrible idea. But I think it's a very good idea to open it up that way, because then you really are serious about what you say about a paper. You can't trash somebody's paper without good reason. You have to be able to defend yourself. I really like it. I kind of miss it. I don't do it anymore, except for circumstances like this. But I think it's not a bad thing to make reviews non-confidential.
Yeah. Well, I think people hide behind the confidentiality and they get nasty.
Wouldn't that be interesting if they did this for all the grant reviews also?
Well, you know who's on the panel. You could figure out who reviewed.
Yeah, but you don't get a transcript of what they said. You get an excerpt from the secretary.
Well, some things need to be secret, like meetings between the Putin. Oh, sorry, wrong thing.
Right.
Those are transcripts that are widely known among the Russians.
One more thing I wanted to ask you. You have the title is Circular DNA Tumor Viruses Make Circular RNAs. So I thought the genomes of these viruses were linear.
Oh,
so what do you?
No, they're only during lytic replication, but during latency, these viruses circularize.
Right.
Otherwise, they would look like DNA fragments, and they would activate DNA damage response.
But in the particle, the genomes are linear, right?
In the particle, they're linear.
Okay.
They're stripped of histones, and then they're packaged under pressure. And if you do a good cryo-EM of a herpes virus particle, it looks like a rope. Looks like a nautical rope that's
a bowline.
nicely.
A bowline.
Yeah, a bowline.
Oh, so we can follow up from last week. So these are naked DNAs that come in the nucleus. They must be immediately coated with histones, right?
They don't have histones in the virus particle.
Right, but when they get in the nucleus,
when they get in,
they pop out. Yep, then they pop out, they travel, I believe, as linear molecules to the nucleus,
right,
recircularize, and then are what's the word, histonellated?
Well, chromatinized.
Chromatinized. There you go.
So I want to know, are they silenced, or because last week we had Steve Goff who said most DNAs, they get in the nucleus, they're silenced because it's not a good thing to have just any old DNA walking in. So just to take a phrase from Lord of the Rings, one does not
simply walk into the nucleus
without paying the consequences. So
you lost me on that one.
I'm sorry.
That's okay.
So what prevents the silencing of capaces and EBV DNA as it gets in the nucleus? Is that known? That's my question.
Well, the answer is actually the viruses want to be silenced. Can you be teleological, or I guess so?
That's part of the normal replication is to be silenced to become latent, right?
Well, if it's latent, then it's not making any proteins that can be processed by the proteasomal machinery and presented as antigen.
Yeah.
And so what we see for herpes type 1 is that it actually does not make any proteins during latency. It only makes a RNA called LAT. And so a herpes-infected neuron that is latent, the virus is just sitting as a circular episome in a G0 arrested cell.
Right.
And it's persisting. Now, if there's a signal that perhaps the virus is being recognized by an immune response or something else is happening, all this intracellular signals that tell a virus to replicate, then it initiates this very complicated and beautiful series of transcriptional cascades that activate all 90 or so genes to be made into proteins, packaged and turned into packaged in virions, and then shipped off as linear RNAs. Got RNA on the brain. Well, I hope not, actually. DNA. So the linear DNAs, they're thought to be made by rolling circle DNA, where there's one point that's comes off, and then the DNA is clipped into unit length linear DNAs and packaged under high pressure, about I don't know how many atmospheres pressure, but it's a lot.
Yep.
And then off they go to infect the next person. For KSHV, it all of these same things, except KSHV and EBV is they're infecting B cells that are not G0 arrested. And so much of their machinery that they have to express during latency has to be able to allow the replication of these closed circular episomes and maintenance of these closed circular episomes during an infected B cell as it replicates.
I'm looking up the pressure. I can tell you that. I just wrote it down somewhere. It's like 800 something per something.
Really?
Pounds per square inch?
No, it's not pound for square inch. But it's not. Phages do even more pressure than that.
Lord.
Yeah, it's a lot of pressure.
Where does that come from?
You have pressure on you.
I mean, how does the pressure...
Packaging process. Yeah.
How does it get generated?
Packaging.
That doesn't answer the question.
Well, as the DNA is pushed, there are motors in some viruses that wind the DNA into the capsid. Well, herpes has a portal that winds it in and pressurizes it. It requires energy.
ATP.
Mm-hmm.
Twix bars. Pat, can I ask you, it's kind of unrelated, but it's the same virus. So we've talked a bit here on TWiV about the potential involvement of herpes simplex viruses in Alzheimer's disease. I'm wondering what your thoughts are on that.
Well, it's a good question. So there's been a recent study that is not looking at herpes simplex because that had been thought about a long time ago, if I remember. But actually, HHV6, which is kind of similar to HHV1, which is typical cold sore virus, and HHV2 is a typical genital cold sore virus. So those viruses, they're possible. The part of the problem is that for HHV6 is that there's an old story that HHV6 might be involved with promoting multiple sclerosis. And there is very compelling data for it, that if you take patients at autopsy who've died of multiple sclerosis complications and take their brains and look at it, you see you can isolate HHV6 DNA and even RNA, I think, from it. But it turns out that a dying brain will reactivate these viruses. So you have to know whether it's a specific thing to Alzheimer's or is it not.
I see.
So these viruses in general are pretty ubiquitous. KSHV is not, but I told you about EBV being roughly 95% of the population. Most people are infected with the other viruses. I mean, we're all chimeric for these viruses. They're living in our somewhere in our cells, in our neurons or our B cells or somewhere, and they are, roughly 50 to 80% of the population has been infected. So then you have to say, well, what's different about a patient who's infected who develops Alzheimer's and doesn't? It's there. It's a real possibility. I wouldn't say it's necessarily high on my list, but it's possible.
There's a trial here at Columbia enrolling at the moment. Take early onset Alzheimer's and give them 4 grams a day valcyclovir for 2 years. And then do a PET at onset and a PET at the end and see if it makes a difference.
That certainly would do it.
Get an answer, yeah.
Yeah.
Either that or you make a lot of antiviral resistant viruses.
They're going to have to be pretty hardcore viruses to resist 4 grams a day.
If they're mutated, maybe?
Okay.
Yeah.
Also, I don't know how much of that gets through the blood-brain barrier.
Right.
So I retract that.
Yeah, maybe that's why they need to give you 4 grams a day. I don't know. All right. Thank you. Thanks for that. All right. Last thing we need to do here on TWiV, picks of the week. I don't know if you remember, Pat, last time you actually had a pick. So we'll let you go last if you want to think of something.
Yeah.
Let me start with Brianne, who hasn't been here for a while.
I have not.
Yeah, welcome back from South Africa, by the way.
Thank you.
It's belated, but...
it's a lot colder here. I don't know why I thought that was a good idea.
Winter here, yeah.
Yeah. Because, in the 80s there. It was wonderful. Very tan. So I found an article on Stat News called "The Herd to the Rescue: How an Invisible Shield Can Keep Us Healthy." It explains herd immunity quite nicely and has a little video explaining herd immunity as well. I rather frequently am asked to explain to my students, my family, random people on the street, the importance of herd immunity, and I really liked the explanation that was in this article, and I will probably be giving it out to many of my friends.
This is nicely done. Yeah.
Cool. Yeah.
Speaking of herd immunity, we have a measles outbreak here in New York City. It's like over 200 cases. It's ridiculous.
It is ridiculous.
They actually have signs in JFK.
Yeah, I heard. Yeah, make sure you're vaccinated, right?
Yep, they do.
Wow. Alan, what do you have?
I have a local attraction. If you find yourself in Western Massachusetts, especially in the wintertime, this place is great. The Magic Wings Butterfly Conservatory. And this is just a delightful place. They've got about an 8,000 square foot facility, most of which is taken up with a huge tropical greenhouse that is full of tropical plants and flowers and butterflies.
Nice.
And also birds, and they've got some reptiles and such on display. Those are in cages, probably so they don't eat the butterflies. But it's just a really, really neat place, and it's warm.
Yes.
You go in, and there's a koi pond and tropical foliage, and it's 85 degrees, and it doesn't matter that it's so brutally cold outside.
They write here, the Lepidopterosaurus in Western Massachusetts.
Yes.
Hey,
Yes, they are.
Western Mass. They copied that from TWiV. Oh, wait, it's not really. Okay.
I think they may predate us, but
Dickson.
Well, this follows up on something I asked before, and that was that I wondered how long it would take us to get a DNA sequence on virtually every living thing on the planet. And we're heading in that direction, and it's getting easier to do, and it's getting faster, and it's getting more accurate, and all these other things. And then I ran across this explanation for the English Science Initiative to sequence 500,000 genomes, but they started with 100,000, and they want to go through Europe to bump it up to 1 million genome sequences. And I think at that point, Pat, I think you can start to answer your questions about, the causality of, just fill in the blank, and you're probably going to find out, but I don't know how many is enough to say something about all humans, but they're going to find that out, because I think given this kind of, projection as to how this is going to pan out, in 10 years from now, you may very well be able to get everybody's sequence done. It's incredible.
This is an awesome project.
It's amazing. Epidemiologically speaking, it's just...
The goal at one point is everybody at birth gets your genome sequenced, right? And then just give it to Facebook, because they're going to get it anyway. They're evil.
They are. They truly are.
Kathy, what do you have?
I have a fun site where you can find out your birthday on different planets. And it's pretty fun. You just type in your birthday and year, and it tells you how old you would be on Mercury, Venus, Mars, etc.
Love it.
And then there's an explanation about, how we calculate days, and day lengths are different on different planets. And that's really just kind of random. And then the transit around the sun helps to define a year. And so for age, probably most people would like to be on Mars. And then if you go all the way to the bottom, then there's a couple of other related things, and you can put in your weight. And I think most people would prefer to be on Venus with respect to their weight.
This is cool.
Yeah.
Oh, man, on Jupiter, I'm only 5 years old.
But you weigh a ton.
Wow.
Look like Humpty Dumpty.
On Neptune, I'd be less than a year old.
Pluto, I'm only 9 pounds.
There you go.
This is very cool.
Yeah.
Wow.
And I meant to do it at the beginning of the year when you had your birthday, and I forgot.
Yes.
So here it is now.
Good.
I'm only 1 year old on Saturn.
Not bad.
Yeah.
Well, you have a lot to learn, young lady.
Next time you're feeling bad about your age, just
That's right.
go there and do it.
Yep.
All right. Mine is an article in the New England Journal of Medicine. The title is Vitamin D Supplements and Prevention of Cancer and Cardiovascular Disease. And this is a result of a rather large study of 25,000 plus participants where they gave them vitamin D or not and looked at them for quite a while. Yeah. And because the question being answered here is whether vitamin D reduces risk of cancer or cardiovascular disease, which of course it's being promoted as a cure for it. And there's no real good clinical trial that addresses it. So there is now.
Yes.
And the conclusion is that it has no lower incidence of invasive cancer or cardiovascular events compared with placebo.
D got an F.
I wonder what GNC thinks about all this.
It may be
they'll just sell you something else.
Maybe that being in the sun does other things, right?
Jesus.
Yeah.
I mean,
I wonder about the relationship to osteoporosis. So,
yeah,
because vitamin D, calcium, etc. Yeah.
Yes.
I wonder if they also did those studies along the way, even though they weren't paid for it, because you could have done that then.
Yeah, they weren't reported here, but one can imagine. I don't know how you would collect those data. Whether I mean, if you have to have a different IRB thing, then you might not do it. You can't just collect whatever you want. Hey, let's do this. No, you can't.
Well, and yeah, I mean, you'd have to do something like bone density scans.
Yes,
it's not trivial.
Yeah. Well, they're not invasive. So
they're not invasive.
They're relatively trivial.
They do cost money, though.
Yes.
25,000 plus people. It's a lot. It's a lot of scans. I don't know.
It is.
So, Pat, you have anything you want to share with us?
Can I share something that I hate, but I think everybody should read?
Yeah,
Please.
of course. Sure.
Oh, good. Okay, so that's the last week's or this week's, I guess. Science Times had an article on North Korea's biowarfare efforts.
Interesting.
And you can find it on the website. And the reason why I hate it is because hate is tense, but dislike it a lot is because first it starts off in the first paragraph. It says that a pound of anthrax could kill off all of humankind. Well, that's true. But some fraction of toilet plungers appropriately applied to humans kill them all off. I mean, it doesn't make it. It's nonsensical. So the reason why I don't like it is because the Times in 2003 also did a lot of reporting on Iraq's biological warfare program.
Yes.
And although that was not the only driver for that, it certainly contributed to a lot of problems. And I think that I think biohysteria is something that's real and it's important to avoid, so that it's not that Kim Jong-il is a good person by a stretch of the imagination. Quite possibly does have biowarfare programs, but the evidence that they provided was not weak, it was non-existent in that article. For example, they suggested that the North Koreans might be working on smallpox because defectors have antibodies to smallpox virus. So would anybody else who's been
probably
vaccinated
have antibodies to smallpox virus?
See that the North Koreans might be using dual-use agents to develop dual-use research to develop anthrax, because there are scientists who published on working with Bacillus subtilis and Bacillus thuringiensis, which are bacillus. They're not the same agent at all, but that's the level of support that they have for a very strong stoking of fear in that. And I just really want to see a more rational, reasoned approach to biowarfare issues, and I don't think that provided.
I love it when rogue nations and terrorist groups take an interest in biological weapons, because they're probably not going to generate anything near as dangerous as they could with, flight training and box cutters.
Well, I did send. They showed a picture of the labs that they thought were present in Iraq in the desert, and I saw it, and I said, "Well," I sent a letter to the New York Times. I said, "Thank God the terrorists are using these, because particularly I hope they're using a very weaponized form that's very lethal, because they'd kill themselves and then we wouldn't have to worry about terrorists, because there's no way that whatever they were showing in the picture could possibly be a biowarfare laboratory, even though that's what the claim was. Turns out that they were weather stations. So there is, as we all know, having lived through that there, I don't think a climate of fear is good about things like that, and I would prefer to see a climate of concerned reason. So,
good luck with that.
Yeah, I agree with you.
But I didn't find a cool site like the planetary.
It's okay. The whole culture, whole industry has arisen that rings alarms when we do experiments, essentially.
Right,
right.
The biodefense industrial complex.
Yes. And the last time this happened was when, the complete synthesis of horsepox, right?
Right.
All these individuals. You've enabled the reintroduction of smallpox. Come on, let's be a little reasoned. Yes. All right, we have a listener pick from Eric Delwart.
Eric, yeah.
Dear TWiV, I'd like to propose a YouTube video called The Molecular Basis of Life. This is a compilation of molecularly correct proteins, RNA, and DNA molecules performing their amazing dances, folding, replicating, and transcribing DNA. Seeing these key molecules of life in motion set to music is startlingly beautiful and enlightening and should be enjoyable for both experts and beginners. Because the diverse methods of replicating viral RNA and DNA greatly outnumber those used by cellular life forms, one can look forward to many more such videos. Yes, but the differences may not be visible.
Right.
There are just 7, actually.
Yeah, you're absolutely right. You were absolutely. So this is great. I looked at this, and it's really cool.
Yeah, it's a neat video. Music's a little loud, but it's a very cool animation.
This is very cool. There's a cell biology video that actually looks pretty similar. I wonder if it's made by the same people. Mm-hmm. I use the cell biology one in class pretty frequently. It's called The Inner Life of the Cell.
Mm-hmm.
Eric was on TWiV a long time ago. Rich Condit and I made a trip out there, and I think we were on study section, so we hooked up with him afterwards. And there's one thing I will always remember he said, because Eric likes to sequence viromes of different environments. He said, DNA is everywhere. It's a real problem.
And it's a real solution.
Yes.
All right, that is TWiV 531. Please listen in on your podcast app and subscribe to us. We'd like it if you subscribed. It helps us to see what our numbers are like. And if you really like what we do, please send your contributions over at microbe.tv/contribute. You could use Patreon. You could use PayPal. Other ways you can do it, you can find over there as well. And questions and comments, twiv@microbe.tv.
You can FedEx a briefcase full of cash.
You bet. Our guest today's been Pat Moore from the University of Pittsburgh. Thank you, Pat.
Thank you very much.
It was very cool, a lot of fun. And when you figure all that out that you wouldn't tell us about, we'll have you back.
Yeah.
Excellent.
Dickson Despommier is at Racaniello.org and parasiteswithoutborders.com. Thank you, Dickson.
It was great. I'm sorry I missed the first part.
No worries.
This was good.
You were teaching at Fordham.
Yep.
How was your first class?
It was great. Absolutely great.
This is vertical indoor farming?
No, it's called Ecology for Designers, but it's actually
Ecology for Designers.
If you want to know what it's about, just go to the internet and type out Fordamopolis.
That's a cool name.
And you will see last year's progress.
What would happen if you called the course Fordamopolis?
I'd have to redesign it.
Really? Isn't that part of the whole shtick?
No, it's the end point, but it's using ecology to get there.
Oh.
Okay. Thank you, Dickson.
Biomimicry. You're welcome.
Brienne Barker's at Twitter as BioProf Barker. She's over at Drew University in New Jersey. Thank you, Brienne.
Thanks. It was great to be back. I learned a lot today.
Are you going to Africa again next year?
I am, but I think it is going to be summer of 2020 instead of in the winter.
Oh. I would do that too then. Did you get into any game parks?
We did. We went to Pilansburg.
Nice.
Game park. We went to a place where some early human remains were found, and I got into a great discussion with my students about, well, what about the first viruses?
Right.
It was a lot of fun.
That's great.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Alan Dove is at turbidplaque.com. He's on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
I forgot to mention that Pat Moore is on anti-social media. He has a website, but he's the only one allowed to go there.
That's right. He tweets himself.
That's right. It's just him.
Auto-tweeter.
I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASV and ASM for their support of TWiV, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
I thought it was cool that you had some tumors that were supposedly EBV negative, but then you found the circular RNAs, and then you were able to get DNA from some of those and go back and show that they really were EBV positive.
That's right.
That was nice.
Yep, that's right. That was interesting because you're working how they determine positivity is they use EBER, which RNA hybridization also usually in situ hybridization. but sometimes it's not that sensitive,
Mhm.
and it can be more sensitive to find the circular RNA instead. So you're right. That was kind of neat.
So we have the circles of obligate intracellular parasites going in RNA circles, the circles of RNA circling back for Moore, viruses in the round, what goes around comes around, circular reasoning, Circ du RNA,
I like that.
and cropped circles.
Circ du RNA, that's good.
That's like Cirque du Soleil.
Yes.
Yeah.
Did you put
I've
that
got
in there?
I did.
Yeah,
That's mine.
That's good.
That's you.
Well,
I
that's
actually I logged in this afternoon to put that one on, and it was already there. So it's good that Dickson already.
Well, you transfected me.
Except it's Cirque. Oh,
I know.
that's okay. But
No,
it's
no.
okay.
For this one,
UV,
Circ.
but this is Circ.
Circ.
And is
Cirque
it
du.
D apostrophe or is it do?
It's do.
It's whatever it is.
But I
let's
don't
do.
know if RNA would be do or not. Cirque
no.
du RNA?
No. Circ du RNA.
No, you have to do it like Cirque du Soleil. But I don't know if RNA is do. I'll figure it out. You guys want
is RNA in French?
Exactly.
it's ARN.
So
It's ARN.
We do not want to do RNA.
Acid nucleic. Yeah, it's ARN. It's ARN or ADN.
No. We have to make it contiguous with
Then it
the
wouldn't
theme
be
of
due.
this one.
right.
Or let's see here. You have to. What did you say, Dickson?
I just said you had to make it resonate with the data that you present, and Circ is certainly part of this podcast.
Right.
Yeah.
So I think
But we're the question here is it do or D apostrophe?
I have no opinions.
Yeah, it's ARN.
That's- right.
It's
It would be
ARN,
ARN,
D
and it would be
apostrophe.
D apostrophe.
So the apostrophe ARN.
No, but then people aren't
but
going
it's
to get that.
Circ
Oh.
And
darn.
it's just not going to rhyme with Soleil.
Right.
Yeah.
Okay.
So we can either screw up the French grammar or choose a different title.
Yeah.
No, well, I mean, it's not even CIRQUE, so it's not
Right.
going to be perfect anyway.
No, it's
So
not.
It's Circ. You want to do Circ D apostrophe RNA? Is everyone okay with that?
I'm good with that.
Yeah.
Sure.
And Pat likes that. So
Oh, do I get a vote?
You do.
Of course.
Oh, yeah.
you like that
I
over?
think that's beautiful.
Is that better than circling back for Moore?
Oh God, yeah.
Okay.
Well,
All right. He
yeah,
doesn't
and
want his name in the title. He's too modest.
Exactly. So
yeah. So, and just maybe we already went through this, but for Cirque du Soleil, it is DU.
Yes.
Yeah, it is.
Right, yeah.
Okay. All right.
So do you want DU RNA? Just
I don't know.
so
No,
that
but when you
people,
say it, it'll come out
Cirque du
different.
Soleil, Cirque du RNA. I don't know.
Right.
Yeah.
because we'll get letters from French listeners saying,
We'd
"What
be
the hell are
glad
you
to
doing?"
answer them.
And just then you can just play in the Easter egg.
Exactly.
So, Pat, do you like Cirque du RNA or
I think that would be perfect.
Cirque du
Because-
RNA?
Yeah, that would be fine. And the French people I know wouldn't complain.